So in case you missed it, I'm Terri Conneran with KRAS Kickers, and 5-year lung cancer survivor. And just, you know, at 5 years, you start getting really excited to be anywhere and starting to really feel the numbers, right? And it's like, okay, that 5 went fast. I'm here for 10. Let's keep going. So I'm happy to hear tonight to bring to you guys Lauren Adams. And Lauren, I foolishly did not put up on my screen like all of who you are and all your degrees and all this other stuff. But Lauren, I had the opportunity to, I hunted her down a little bit is what happened. There's this thing called the KRAS Initiative that she's going to go through with us a little bit. And she was a presenter there. I think it was 2 years ago. And I saw that she gave her presentation. And so I reached out to her and connected with her there. So Lauren, if you would be so kind as to just tell us a little bit about yourself and then just kind of go into your presentation. Welcome tonight.
Thank you so much. Thank you guys for all being here today. So I am a senior graduate student at Northwestern University, studying to get my Ph.D. And I am looking at my research with KRAS, and I'll be talking a little bit about that today. As well as the KRAS initiative and things like that, and how that's really impacted me as a researcher in the work I've been doing. I like informal kind of presentations, so feel free to interrupt me, send something in the chat if you have a question. Like I'm more than happy to answer any questions. So with that,
No.
Did he force you?
just all right, can everyone see my slides okay?
Yes, perfect, thank you.
All right. So the two kind of main topics that I want to talk to you guys about tonight is first, what does a KRAS mutation mean at the molecular level? So I'm going to try to do my best to break down all the jargon and kind of what does it actually mean when there is a KRAS mutation inside the cell. And then at the end, I'm going to be talking about the NCI RAS initiative, what that is and what it does and how it's impacted me as an individual. So to really be able to explain what a KRAS mutation means, I'm going to have to give a little bit of background. So I'm going to try to avoid any of the typical scientific jargon, but if I slip up and I forget, please stop me, ask me. So the best place to start when trying to explain this is going to the central dogma of biology. So I think probably everyone is familiar with DNA at this point. And so DNA is your genetic information, which tells your cells basically how to create a lot of the different cellular components that make the cell operate. And so there are a number of steps that actually have to happen to be able to go from your DNA and genetic information to the protein level, which is where KRAS is important. So an analogy that I really like for this is that I've heard is DNA is like a cookbook that you have to keep in your living room. And this is like before cell phone technology. And so you'd have to go to your living room and write down on a piece of paper what the recipe is that you want to cook for the kitchen. And so the DNA in this case is like that recipe from the book in your living room. And then the cell goes through this middle step, which is at the RNA level. And that's like you writing down the recipe on that piece of paper. And then finally, the cell uses that written out recipe, middle step, to then kind of code or tell the cell what to make for your protein. And so this middle step here, which is the RNA level, every 3 base pairs, so these are showing every 3 pairs of these letters in that code actually tell the cell this is the part of the protein that you need to make. And so then you get a bunch of those added together and you get your total protein. So what actually is a protein. It's a chain of amino acids. So here on the left, these are just chemical structures, but essentially there are 20 different amino acids that your cell can make. And they all have different chemical structures and different chemical properties that influence how the protein behaves. So basically this is like a string with a bunch of different amino acids that are all attached to each other. And that's what we call our primary sequence, is when we just connect them all up in kind of like a line. But then we can add complexity to this, and we get all of these different structures that the protein can then take. So there's like secondary structures, which include different hydrogen bonding, but then you can get more complex structures where these chemical bonds are interacting with each other. There's all these different molecular forces. And this is really important, this structure in 3D space for how the protein actually behaves. So that is very important. And then there's also an extra level where multiple proteins or the same protein together can create a giant complex and behave as one kind of unit. So there is a lot of
Can you go back a quick second?
Definitely.
Okay. So it's primary, then secondary, then tertiary, quaternary. Did I say that right? Okay.
Yep.
And so quaternary is basically the primary is like the one, secondary is taking the two together, right?
Yeah. So primary is just like the basic string of the chemicals.
Okay.
Secondary is kind of a simple 3D orientation that happens through hydrogen bonding. It's kind of giving like this helix shape.
Okay.
Tertiary then is like taking the secondary structure and then further folding it into all different kind of shapes, and that is dependent on what kind of chemical structures you have within that primary sequence.
Okay.
And then the quaternary is sometimes you can get multiple of the same protein that combine into one giant complex and operate together.
Okay. And so it's basically, you know, you're getting up to like a group of pretzels, right? It's a group of pretzels, like one less is like one pretzel, right? And the other one's like a piece of it, and then the other one is like just like what you started with the original, right?
Yes, exactly.
Okay.
So then there's some terminology when it comes to KRAS mutations, and since you guys are part of this organization, I think you are probably familiar with hearing these, but what does it actually mean in terms of biology? So sometimes a KRAS mutation is referred to at the DNA level. So you might see something like this first bullet point where it's like C.183A and then this arrow to T. So what does this actually mean in terms of like the biology behind it? So this is a shorthand for saying that the DNA at base number 183, so that would be an A in the letter code for DNA, that turns into a T. And so why is this important is because if you change even just one single DNA base pair, that can then change what your protein looks like. So at the protein level, in terms of terminology, what you would see is something like this, G12V mutation. So this is shorthand. This G and the V are like one letter codes that stand for specific amino acids. And then the number tells you what position they are in that sequence. So for example, for the G12V mutation, the amino acid named glycine, which is abbreviated by G, at position 12, so it's number 12 in your sequence, turns to a valine, which is the letter V. And so why does this matter when you have one amino acid turning into a different one? So right here on the screen, I'm showing you guys a structure of what KRAS would look like if you could see it with your eyes. This is achieved through different scientific techniques, and this is just a representation of what you would see. But I kind of highlighted the important parts here. So this is position G12. So for G12B, G12D, G12C mutations, there's position G13 shown here, and then Q61. And these are some of the most common sites of mutation in KRAS. And why it's so important is because these three amino acids really help KRAS perform its chemistry in the cell. And so on the right here, this is a kind of representation of KRAS as it would be behaving in a cell signaling pathway. So when KRAS is behaving, it helps the cell tell it to grow and proliferate. And we have this in all of our healthy cells. This is something we need to be able to have our cells to be able to grow and function properly. But what happens is these mutations at these sites lock KRAS into an inappropriately active state. So it tells KRAS to keep signaling in this pathway and to keep growing and keep proliferating, and it shouldn't be stuck in that constantly active state. And so that's why mutations at these sites are really critical for KRAS function. Another thing I wanted to talk about is HRAS, NRAS, and KRAS. So what's the difference? Everyone here, I think, is probably most familiar with KRAS, but I'm sure you may have also heard about HRAS and NRAS. And so essentially HRAS and NRAS are the equivalent of protein siblings to KRAS. They're very similar in their amino acid sequence. So down here, I have this figure shown. So what this is showing is that between amino acids 1 and 85, these HRAS, NRAS, and KRAS are exactly the same in terms of their amino acid sequence. And even between amino acids 85 and 165, they still have 90% the exact same sequence. And you really only get any variation in your sequence of amino acid at this this end portion here between 165 and 188 in terms of amino acids. So in a lot of ways, HRAS and NRAS behave similarly to KRAS, but even though they have a lot of similarities in their amino acid sequence, they can behave very differently. So one key way that they can behave differently is how they're modified. So the cell can add these additional chemical modifications onto the protein. And what that does is that can alter how what different cell signaling pathways HRAS and NRAS can signal through. It also can dictate what proteins in the cell that these proteins, HRAS, NRAS, and KRAS can interact with. And these are all important for helping your cell function. And then there's also studies that show that the expression of these genes alters over time in the cell life cycle, as well as at different tissues. So, for example, your heart tissue may be expressing different levels of HRAS, NRAS, or KRAS than your colorectal tissues. So another important note at the molecular level is that the context really matters. So a KRAS mutation, let's, for example, take G12C, does not always behave the same as if that G12C happens in, say, your lung cancer versus pancreatic cancer. And so this is important when looking at therapies and things like that, because ultimately the tissues that these KRAS mutations occur in, they have a lot of other different proteins and a lot of other genetic information that's going on that regulates those tissues differently. And all of those can influence the impact of a KRAS mutation. So why would a KRAS, why is it really important to know what your KRAS mutation status is? So there are a lot of studies out there that show that a KRAS mutation can impact the interaction with other proteins in your cell. So a G12C mutant KRAS might be behaving and signaling differently than, say, a G13D mutant KRAS. And in addition to that, certain mutations are more common in specific types of cancer. So really G12C is a great example because it's one of the most frequent mutations for KRAS in lung cancer. But when you look at pancreatic cancer, it makes up a very small percentage of KRAS mutations. Instead, a mutation such as G12D is a lot more prevalent in pancreatic cancer. So it's important to really know what mutation you're dealing with. And then, as you guys are also probably aware, is that the KRAS mutation status impacts what kind of therapies you're receiving. So I think from your previous lineup of speakers, there's been people that have talked about therapies such as KRAS, and those are specific to certain mutations. So that's really important, as well as even just other kinds of therapies, even if they're not specific for KRAS. An example of that is if you have a mutation in KRAS at position 12, 13, 61, any kinds of those mutations, then doctors probably won't be putting you on antibody-based EGFR inhibitors because those have been proven not to work in those cases. So it's really important to know at the molecular level how things are behaving. So what does KRAS need to mean exactly? So I am a PhD candidate in the Kelleher Research Group at Northwestern. And in my lab, it's actually a very analytical chemistry driven lab, but I study KRAS as my application. And so the best way to really kind of explain what I do is that I weigh KRAS to determine what mutations are present at the protein level, and then what kind of chemical modifications the cell puts on that further. And so a really good analogy for that is, say we have a scale here. This is something you would weigh yourself in the morning, and you have a person. And so essentially what we do is you would weigh yourself, or you would know what your weight would be without any clothes on or anything like that. So you have that base weight. And then say you have a full piece suit with shoes and everything like that, but we know what the weight of each of those pieces of clothing or each of those shoes are. And so we would then have the person step onto the scale, and we would measure their weight with all of those clothes on. And then from there, we could deduce what they're wearing because we know the weight of each individual item. And so this is essentially what we're doing, but with KRAS. And I use a much more sophisticated scale per se. It's called a mass spectrometer. But essentially, if we really boil it down, this is what I'm doing: is I know what the mass or the weight of KRAS is without any mutations or any modifications. And so I use that and then measure the mass of what KRAS is actually in, say, cancer cells or tumor tissue. And then I can tell what kind of mutations and modifications are on there. And so this is really important because these modifications can dramatically change how KRAS is behaving in the cell and dictate how it is signaling. So we really want to understand the relationship between, say, mutations and different modifications. So how is KRAS behaving with these mutations as well as with modifications? So this kind of leads me into the RAS initiative. So just to give a little bit of background on the RAS initiative, in terms of history, is in 2011, an advisory board for the Frederick National Lab for Cancer Research wanted to identify and undertake important and ambitious projects in cancer research that would be difficult to pursue without an orchestrated effort. And so it was very much an initiative of, let us try to tackle some of the hardest problems in cancer that we have not solved and really dedicate time and effort and money into trying to solve those. So then in 2013, that's when the RAS initiative launched, because RAS mutations were seen as one of the biggest unsolved problems in cancer research at the time, because we had known about KRAS and HRAS and NRAS for many years now, for over 30 years, and a lot of the traditional ways of targeting cancer and proteins involved in cancer just were not working for KRAS. So we then, the RAS initiative then mobilized the community to develop these ways to understand target mutant KRAS cancers. So what kind of the RAS initiative is it's this hub where it connects all the different members of the RAS community in terms of researchers. So this is their figure. They have their Frederick National Lab for Cancer Research, which is in Frederick, Maryland. And it's kind of like the base or the home base for all of the research that's going on. But it's really a lot more than that, because there are collaborations in biotech, in pharmaceutical companies, as well as academic research labs. And so essentially it really helps with all research that's focused on RAS, and it helps with funding. It will give us chemicals and supplies. It has extensive protocols for researchers that helps them with their research, lots of collaborations, and then it just provides access to experts in the community at large. So it's a very collaborative environment, connecting a lot of the top RAS researchers. So I have personally been impacted by this, and in many different ways. So one of the biggest ones was that the RAS initiative helped fund my research project that I've been studying for the past 5.5 years. And so while we were on this funding, I would have monthly update meetings with other researchers that were actually at the Frederick National Lab. We would provide annual reports. And then also, more importantly, they gave us access to critical reagents. So this means they would ship me cell lines. They helped me access tissue samples, as well as different materials that were really critical for me to be able to do my research. And so by them sending me these items, it saved me years of time. And that's no exaggeration. It saved tons of money because I wasn't trying to do all these things myself again. And it just was really helpful to have these resources so that I could actually focus on the questions that we were interested in terms of KRAS. So an example is they gave us access to a cohort of colorectal cancer tissue samples. And this was really important because I can study cell lines as much as I want, but those are always kind of an example, and they're not always the best reflection of the complex environment in actual cancer. And so,
Hey, can I stop you for a second more?
Yeah, sorry.
or before we started, I thought it was sounded a little garbled. We're having a little bit of feedback from your talk, where you sound kind of garbled. It's hard for us. Are you on a separate microphone, or would you mind calling in so we can kind of get a clearer?
Yeah, let me call in 1 second.
Yeah, no worries. That's okay. We will amuse ourselves. Fortunately for you all, I do not sing or dance, and so I will not be performing this evening. So you're welcome. But and then if you are part of my family, you heard me sing happy birthday or some other such nonsense, you would be applauding that mere fact that I will not be singing or entertaining you this evening. So there is that. I did not know anything about this whole RAS initiative thing until I found out that I had KRAS. And so this was brand new to me. I don't know. Annie asked the question, does the RAS initiative need more tumor samples that are more rare? I have no idea. But I can tell you that the KRAS Kickers and I were invited to present a poster at the RAS initiative. Yes, much better. And that is going to be one of the questions I posed there, Annie.
All right, 1 second.
No worries. That's okay.
That was so much better.
Yeah, right.
Yeah, thank you.
Beforehand, they said it was like, I was having a hard time understanding. It's like, okay, well, maybe it's just my speakers going out. So
Right.
you're saying something. Glad to see
All
you.
right.
Worried.
Yeah.
Okay, right.
Is everyone able to hear better?
Yeah, much better. Okay, Lauren. Okay.
Okay, cool. Do you want me to repeat anything, or is
Might just
want me to
start
keep
at the top on this, what this slide is, and then, so we can get you.
all right, awesome. So one of the really important ways that the RAS initiative helped me with my research was that they gave us access to a cohort of colorectal cancer tissue samples. And this is really important because I had access to a lot of cell lines, but those are kind of more of a model system, and they're not always reflective of what's actually happening in the tumor environment. So we wanted to really get a good idea of what is happening at the patient level. And so an example here is, this is data from my research, and this is showing different tissue samples with different KRAS mutations. And these
We're not used to looking at this. So if I can just pop you for a second. Okay, so the numbers below, like it'll say on the bottom, it'll say like G12V in parentheses, but underneath that, it's got like a number. What does that number represent?
Yeah, so each of these numbers is like an ID that is given to the tissue sample. And so this is to help us be able to identify these samples and to be able to keep track of this as a resource, especially because we do not get any information about where these samples came from to protect patients' privacy. So this is a way for us to ID each sample. And then in parentheses, I have the KRAS mutation that was identified in each of these samples. And so in these black bars, what this is showing is the levels of mutant KRAS protein in each of these samples. And so 100% would be total KRAS protein. And each of these, so for example, this first tissue sample with a G12V mutation has about, I would say, 38% of mutant KRAS protein in there. So this is really important because our method was one of the first methods that allows us to look directly at this in a really good way. And we got to see important trends. So in a lot of the samples, the mutant version of KRAS was about the same as the wild type or non-mutated. But there's also some outliers that we found were kind of interesting. So these last 4 tissue samples shown here, they don't have any black bars, which means we saw no mutant KRAS in them, which was surprising because they have KRAS mutations at the DNA level. So it was really important to be able to get like this first view into how the mutant form of KRAS is being expressed in the tissue.
Okay, let me ask you a question, okay? Because we're not used to looking at this stuff, and so this is kind of unfamiliar territory for us. So those numbers, those identifying numbers that are right next to like the G12V, the G12V would represent what the mutation is, and the O1C, whatever, something, 08, that would represent like a de-identified patient's pathology. Is that right?
Yeah, exactly. Because we don't get any information about the patients to protect their privacy. And so this is a way for us to be able to communicate results and be able to track this back to different resources and things like that. So you're right, it is a de-identified ID number.
Well, that gives me a little bit more confidence as far as like as a patient, if they're taking something and they're doing some study on it. I mean, this is not, I mean, it's just some number out there. It's not like my social security number or something. Okay.
Yeah, exactly. They do a lot to protect patients and things like that. We have to take special courses and go through lots of documents to sign that say we're not going to do anything bad with any information. So, but yes, overall, this was really helpful for us to get to see what is actually happening in a tumor environment in terms of how much mutant KRAS is actually there. And another really important way that they helped me was they provided a genetically engineered cell line. So this saved me years of time, and I'll explain this. So this actually saved me years of time because this engineered cell line was already at the NCI RAS initiative. They've done a number of steps to make sure it was validated and a really good resource. So it saved me lots of time, and it actually enabled me to pursue one of my own scientific hypotheses. So I'm showing you right now is from one of the tissue samples. This is actually a sample that did not have any KRAS mutations. But this is an example of data that I would get. It would make no sense to someone outside the field. But essentially each of these peaks that I'm showing you represents a signal for those proteins that I'm weighing. And so I've highlighted this one particular one that's really high up here by this number 23 plus. And what's really significant about this is this was a completely new form of KRAS that we had not seen in all the years of research. And it was really important because of why it was different. So it was missing one of its amino acids at the end. And this form of KRAS was found in 18 of our 34 tissue samples, and it was very present. So it was, to us, this was a sign that this is very important, and we should figure out how it's behaving, because this form is very different than what we would expect to see. And so I was able to test my hypotheses. So I'll walk us through each of these pieces of data. So right here on the left, we're looking at cell lines, which are expressing KRAS. And so this green here that you're seeing in these panels, anywhere that you see green, that is a signal for KRAS when we're looking at the cell. And then in this middle set of panels, when you see this purple color, that's just telling you where the cell membrane is. And then at this end, we're just overlapping the two images so that you can see how they relate to each other. So in the top here, this is the normal form of KRAS that is not missing any amino acids. It's not mutated or anything like that. And what we can see is that most of the KRAS is hanging out at the plasma membrane, which is what we would expect to see. And so KRAS has to hang out at the plasma membrane of the cell to be able to signal properly through those pathways that tell the cell to grow and proliferate. But what we're seeing is when we look at the form of KRAS that's missing that amino acid, it is not able to associate or hang out at the plasma membrane. It's just all over the cell. And so this is really important because we thought after seeing this that KRAS in this case, when it's missing the amino acid, it can't signal like it's supposed to be signaling. And so on the right, this is, it's a lot of jargon, but let me just break it down for you. We have this abbreviation WT, which just means the unmutated regular form of KRAS. And then I have the C185 star. This just means that this is the form that's missing that amino acid. And then we have this PAR, which this is just cell lines without any kind of manipulation. This is our like baseline. And then these three conditions you can ignore for now. These are just controls to show baseline. And so essentially what this graph is showing is how much cell signaling is going on in these cells. So I inserted DNA that will tell the cells to create either the unmutated normal KRAS, or it tells the cell to create the form of KRAS missing that amino acid. And so what we saw was that the typical form, unmutated form, was behaving normally. It could signal really well, and we measured cell signaling to be near normal levels. But when we looked at the new form that we saw that was missing that amino acid, it could not signal through those pathways that we'd expect it to. And it showed basically the same levels of signaling as just our baseline controls. So this was a really important finding because this is showing that this form of KRAS that was really prevalent and abundant in actual tissue samples can't behave normally and it can't signal normally. So at this point, we don't know why this is happening in these cells, but it may suggest that there's some kind of like feedback mechanism where the cells are trying to stop or slow down the growth in a manner because of the mutations and things like that being in the cancer. So we think this might be some kind of regulatory mechanism. But this was a very cool novel finding, and I would not have been able to do any of this if I had not gotten those cell lines from the RAS initiative. So this is a huge help, an example of how they give you resources to do your research.
Okay. And I was going to say, okay, now that how it impacts you as a researcher and how that impacts us as patients is this is the start of where it starts leading towards potential treatments and potential FDA approved drugs and clinical trials and stuff.
Yeah, exactly. Because there has been a strategy in the past to prevent KRAS from associating with the plasma membrane. But past therapeutics that they've tried to develop for that actually have failed because KRAS had found a way around that to kind of avoid the mechanism of that therapeutic. But if we can figure out why this form of KRAS is happening, that is really a significant therapeutic potential because you could exploit this and figure out how to get more of KRAS to be missing this amino acid. and to not be able to signal properly in cancer cells. So that could be a very significant therapeutic potential in the future. And as Terri had mentioned, we actually met a couple years ago or so through the third RAS Initiative Symposium. So this is yet another way that the RAS Initiative has impacted me. And it's every year or so, they get together and they have all of the best RAS researchers around the world get together, and they present the most cutting edge new research and data in the field. So this is a really important opportunity for people to share research in progress and to be able to further their own research. Just from experience, it really helps to be able to talk to people in your field as a researcher and say maybe you're struggling on trying to get an experiment to work or things like that, or you're kind of hitting a roadblock in your research. You'd be amazed at how sometimes like a 10-minute conversation with a fellow researcher, they're like, oh yeah, I had that same problem. This is all you have to do. Or, oh yeah, maybe you should try like this certain thing and it'll work. And so many times it helps you like problem solve. So these kind of events and gatherings are really important. And kind of along the same vein is the RAS Initiative has a RAS Dialogue blog, and it's just a very informal way of researchers to give a short description of their ongoing research. And so I can log in about every once a month, and I get to see some of the cool stuff that researchers are doing in the field before they have to wait and get it published. So this is something that I was able to participate in as well. With that, that's the content that I had for everyone today, but I'd be happy to answer any questions. And then I did want to plug for Terri, the KRAS Kickers Connect coming up here in September, and she can talk more about the details of that. But thank you guys for your attention.
Okay, thank you so much, Lauren. If anybody has any questions, like just like pop them in the chat, or just like give me, I don't always see everybody moving around. I'm gonna go back to full screen so I can see the whole gallery of people. So if you have a question or something, just like give a wiggle of your hand or something of that nature, let us know. But the RAS Initiative, you know, is as much as like when you're saying, oh, this is impacting you, okay, the way this is impacting us is this is like what's going to potentially be leading to treatments for us, right?
Exactly.
We're not going to be here without, you know, tomorrow's without treatments for tomorrow. In fact, when I had reached out to Lauren, she mentioned to me, and I hope you don't mind me saying this, she mentioned, she's like, uh-oh, what? She's like, I'm the first KRAS patient she ever met. Because even though you may be talking to somebody, it's not necessarily like they know that. And it was really quite an honor for her at her, I don't know, what would you say at your level, to be doing a presentation like that at that high level of a forum. And so I applaud you. I mean, you're definitely an up and comer. And so, you know, I think that's pretty cool. Will you be going this year?
I am unsure yet because it is in person. I'm not sure if I can travel for it, but I would hope so because it is a really cool event, and it'd be nice to get to go to Frederick National Lab, check that out. I have a couple of colleagues over there, so it'd be nice to see them as well.
For sure. Well, like I had mentioned earlier, is I was invited. I met the people that are in charge of the whole RAS Initiative. And if you look at that one chart that you've got where it shows the whole hub and all the different pieces and pharma and this and that, and in fact, if you pull it back up on the screen, if you look at all those different pieces, the one part that you kind of see that's missing, like there's this big hole here, is there's no patients. And that's kind of where the KRAS Kickers come into it, right? Is that there's not another group that is RAS specific. And even though we went with KRAS Kickers because it's usually what people get diagnosed with, there are a smatterings of NRAS and HRAS patients that do kind of like cross over our paths. So it's kind of cool that we have that opportunity. And so keep an eye also, because we're going to be featured in one of those blogs that you mentioned, to kind of like let them know that we're out there. So that's kind of cool. Let's see, Annie made a comment. Dr. Lori also is interested in those K117 mutations. Okay, the fact that Annie knows that off the top of her head is a little disturbing, but cool. I have no idea what any of that meant specifically, and I'm not really sure where that falls. It sounds like it's very cutting edge. It's a little too bleeding edge for what it is we're going to be seeing, but it's up and coming for like next steps. Is that fair? Is that fair?
Yeah. Yeah, I know K117 mutations are less frequent than the typical, G12, G13 ones, but they are still prevalent enough. So I would say there's so many ongoing therapies being developed and research being done, targeting KRAS from so many different ways that I wouldn't be surprised if someone is working on a specific inhibitor for, say, K117 mutation.
Okay, cool. Okay. Does anybody have any questions? I'm looking through my screen here to see if I've got like any wiggling or anything going on here. And there's my gallery. Okay. Well, Eric is probably the only person that apparently is not in the witness protection plan this evening, because everybody else is like just taken to hiding. And I noticed earlier you had a really cool looking bowl, just that you had like a really neat looking dish. So yeah, it is like just neat looking. I don't know what that is. That looks very, very kind of like funky. Very cool. Does anybody have any questions oh, Maureen's over there. Do you have any questions, Maureen? She's like, no, just saying hi. Okay. Okay, cool. Okay, cool. Lauren, this is your opportunity to kind of like just like hop in here too, because you have not really joined with our group. Do you have any like thoughts or questions? It's kind of like a researcher having an opportunity to talk to like a patient group.
Yeah, 1 second. I do see a question here in the chat. So is there a clinical trial network the RAS initiative belongs to? Are patient advocates that they invite to help review new trial protocols from the patient's perspective? Okay, yeah, so the RAS initiatives themselves, I don't believe that they do any kind of clinical trials themselves. So I do know that the RAS initiative at the Frederick National Lab is developing like early phase therapeutics and drugs and stuff like that. And likely what they would be doing is they would work on early phase parts, and then they would collaborate with hospitals and research institutions that are, or say biotech or pharma companies that are better able to have that infrastructure for clinical trials. In terms of like patients being able to advocate for different, I guess, different protocols for the trials, that I don't know. I think you could probably always advocate for certain approaches, but I don't know how much wiggle room there is for changing things, especially with the early phase clinical trials and things like that. It's really important that they're structured a certain way to be able to pass, say, FDA approval. So I don't know how much input they can get from a patient's perspective, but it would be interesting to reach out to someone who's more involved on that side of things, see what they think.
Yeah, because that's not really where the clinical trials are coming down through. We do work with like some, quite often I'll get different, like maybe a pharmaceutical company or maybe a clinician will reach out and ask for participation. And so as those opportunities like come out, I might reach out to somebody. If it's kind of like in your area of scope. So if it's not anything that you're familiar with, I want to reach out. So you're on my list as far as volunteers, Susan. So you're already there. See, Annie had a question. Does the RAS initiative need more tumor samples and they're more KRAS rare? I don't know. I think that's probably a really good question. I'm sure that somebody needs something more somewhere. And that's going to be one of the questions that I pose to them there.
Yeah, that's actually a really important point. So I want to say that, so when we got our tissue samples from the RAS initiative, it was a huge deal to go through. Like it was like a year-long process where we had to advocate like why we were the ones that needed those tissue samples for our research. We had to provide presentations, we had to fill out extensive paperwork. And then even from there, we only got a subset of samples that we requested because they are such a valuable component of research. Everyone wants them because they're so important for research, but there's limited resources. So I would say like having looked at the list of like available tissue samples in their kind of tissue bank, there'd be maybe like one or two of the rare KRAS mutations out of all of those. So I think that if we, and this extends also to like say cell lines, some of these rare KRAS mutations, there's just not many cell lines even to use as a model. And so I think that's why a lot of the research is biased towards the heavily mutated forms because it's more readily available for research and things like that, whereas the more rare mutations are hard to get access to, you know, the things you'd need to actually be able to study them. So it's a great question. Let's see.
Let's see.
So
go ahead.
So yeah, so how did I decide to research KRAS? Did you make a personal interest? So I kind of was interested in KRAS because when I joined the lab that I was in, I really wanted to have research that had that was applicable to helping understand like human health and things like that, and hopefully something that could translate research into furthering, say, patient health or therapeutics or stuff like that. And the KRAS project was one of the probably biggest research projects in the lab that did that. So I thought it'd be really interesting to be able to get to examine this issue. It was pretty a novel take on it. I've known a very long-term KRAS collector. That's a great question. So I have known a few very long-term KRAS colorectal cancer survivors. Is anyone looking at their mutational profile? That I don't know, but would be really interesting to look at because, you know, colorectal cancer doesn't have a great 5-year survival rate. And so being able to see patients and research what's different about them, that when they live far past the expected time, I think that would be really valuable because maybe there's something special about their genetics, maybe there's something going on that we don't know about, and being able to see that would be really important to maybe understanding a therapeutic that could help more people. That's a great question.
I don't see any other questions. Does anybody have anything that they just want to like jump in on? Don't make me sing, people. No, don't worry. That's not going to be a problem. Okay. No. Okay. Well, I really appreciate your joining with us tonight, Lauren. Did you have any questions that you want to like pop out to us? Just kind of, you know, you're just kind of curious. We have a pretty open group of folks here, and I know most of the people online, so, you know.
I guess, what is there like a general consensus of what you all are interested in the most in learning about, like KRAS? Like, do you wish that you could talk to more researchers? Do you wish you could understand more of, say, like what's going on the molecular level? Like, how well is all of this stuff like communicated to you as patients from the scientific community as a whole. That's something I'd be interested in.
That's exactly why we do these. Okay, is when is we don't know the answers and we don't get the information. And so even when a researcher or a doctor presents the information to us, right? Even though they think that they're not using nomenclature, a lot of times they really are, because it has more specificity than you think it does, right? And so I think it's that part of it. And what you're doing in some ways, because it's been so de-identified down to the cell lines and stuff, okay, for good reason, because you don't want to know that you're looking at, like, you know, your cousin's like tumor or something, right? I mean, you don't want to know that. And as a patient, I want to know that it's not my social security number floating around there, right? And so you need to have that level of safety in the middle. And so, but sometimes when it gets de-identified down, that's kind of like where I think we lose the sense of it's a real patient, it's a real person, you know?
Exactly.
Does anybody else have any thoughts on that? Oh, okay, wait a minute. Oops, I'm sorry. I see questions popping up. Okay, in your presentation, you mentioned the Mirati G12D drug. Is this available? No, this drug is not available for a patient that has that right now. I'm also a G12D patient, just so you know. I didn't see who wrote it. But it's not there yet. It has not opened up a clinical trial, though they said, last I heard, probably by the end of this year. And so it's definitely something that's on the radar, and I stay right on top of that, because there's a shortage of G12D clinical trials. They're starting to open up. It's very, very small, a lot of phase 1 clinical trials. And in the phase 1 clinical trial space, you're looking at very small groups of patients, like around 20 maybe in many cases, sometimes less. And it's not like it's all over the country. It may just be at 1 or 2 centers, just very specific. Okay, Eric had a question. Can you talk a bit about the difference between targeting an upstream signaling element such as RAS versus a downstream one such as ERK and mTOR? And again, see, you're going to see, Lauren, like Eric's question is very insightful. Okay, very specific, very aware as far as like a lot of us know where the pathways are, and the EGFR leads to this, and right, and BRAF is, and other ones, but we don't start out that way. It's not like we learned this in school or at patient 101. But if you want to answer,
yeah, exactly. Yeah, no, that's a great question. And so, yeah, it's also a very complicated one. So there's a lot of different inhibitors in that same pathway. And it depends on what is mutated in that pathway. So for, say, a RAS mutation, you would preferably want to target RAS itself when it's being overly active versus, say, like an ERK inhibitor. But at the same time, there's also a lot of ERK mutations and things like that that are frequently happening in cancer. So those might be better options for targeting if that's your case. I know that sometimes when they try to use, say, ERK inhibitors alone, there's feedback mechanisms that the cell can kind of undergo, and it basically allows the cells after a certain amount of time to kind of circumvent that whole therapeutic option. So I've also seen combinations of these different therapies being used because they're really just trying to knock out everything in that pathway. So I would say it's really patient dependent, and that's something that a doctor would probably be best at deciding what would be good for a specific patient, what combination of therapeutics.
Oh, I see another question. And yes, Annie, I totally missed your question. Apologies. You've heard of RAS amplification being related to overall survival in colorectal cancer. Yeah, we didn't even talk about amplification at all. Dr. Laurie's research. Is there a way to tone down amplification besides a targeted therapy? I don't think it's a dumb question. It's a dumb question. I know the answer to it, and I don't know the answer.
Yeah, no, it's definitely not a dumb question. And I think researchers are probably looking at that. It would probably have to involve some kind of like gene therapy in terms of you'd have to limit the expression of KRAS at the DNA level or that RNA kind of recipe writing down level. That would probably be the best strategy. And I actually don't really know like what stage those kinds of therapeutics would be at. So it's a really great question though. I'm sure someone is looking into that.
Terri, can I talk?
Yeah, please do.
I just wanted to say thank you so much for coming on and for speaking to us and for researching this. I know when I had my testing done, it came back, you know, that I had KRAS. Someone had said it's untreatable, and I'm like, or undruggable, not untreatable, undruggable. And I was just like devastated. I was like, oh my God. And just joining these groups and listening to doctors and researchers, and you know, it's hope. There's hope out there. People are researching it. Things are being done. There is a drug on the market now for, you know, KRAS-C. It just gives us hope. And I want you to know that I really appreciate the fact that you are doing this work. I just want to say thank you.
Thank you. That means a lot. And I really appreciate hearing all of that. And it is a really great time in terms of RAS research. Like the past 5 years, there's just been lots of progress. So I think it's great. There's lots of hope, especially in these past 5-10 years. So keep being awesome. And thank you for coming and listening to my talk today.
Yeah, I totally agree with what Maureen said. You know, it does make a difference. And, you know, we don't get to see like the people behind the research, the same way you don't get to see the people behind your cell lines, right? And although you may not be looking at my cell line, right, it represents a piece of me and it represents a part of our community. And so it's a big deal to us, you know? And I think that in many ways, you guys are the unsung heroes, okay? But because even when, you know, one of your research projects goes poorly, meaning it doesn't work, right? You know, it's a great hypothesis, it seemed perfectly logical, and then it just didn't work. That seems like it's bad news, but it also tells us what's not going to work. And I'd rather it not work in that cell line or in that mouse or whatever than not work in me, right? Or my kid, or
Exactly.
whatever, right? And so it's still progress, they're going in the right direction, you know? And it does make a difference. And being able to have, you know, those brilliant minds coming together really pulls the energy that we need. So keep doing what you're doing. Okay. We know that you see, after you finish up, you're gonna be looking for a job. So like, you know, shout out, Lauren's gonna be looking for a job, what, in a couple of months here, right? You know, or that's what you want to do. Do you want to work in pharma? You want to go educational?
Definitely want to go pharma route. I think it'd be really cool to work on the side of developing therapeutics for patients. So that's the route I want to go.
Okay. Well, you know, you never know who's going to hear these, and so you know, make sure that you're tied into that, and you know, on your LinkedIn and whatever. So, but we do appreciate your being here.